All right, get ready, because today we're doing a deep dive into DNA. You requested a chapter. It's like DNA Technology 101. And wow, this stuff is amazing. We're talking gene editing. We'll be talking about cloned organisms. So many things to cover. Let's just jump in.
I agree. This chapter really covers a lot. It starts with like basic DNA structure and then goes all the way to applying it in medicine and forensics and agriculture. It's a lot to unpack.
You're definitely the expert here, so where do we start? Like, what do we need to understand first before we can get into everything else?
Well, you have to be able to read life's blueprints before you can tinker with them, right?
Right.
So that's where DNA sequencing comes in. Think of it like you're trying to decipher the instruction manual for building and running an organism.
So, like, figuring out the order of all the A's, T's, C's, and G's in the DNA code.
Exactly. The first methods for doing that, like the dideoxy method, were really tough. But then next generation sequencing came along and it really changed things.
Oh, yeah, I've heard of that.
Yep.
Like, they can sequence tons of DNA fragments at the same time.
Right. Yeah. It's called sequencing by synthesis. And the speed is incredible. We can sequence something like 2 billion nucleotides in just one day now.
2 billion. Wow, that's just mind-boggling. Like, how do you even make sense of all that data?
It takes a lot of computing power, that's for sure.
Yeah.
But the insights are completely changing things. We've sequenced the genomes of so many organisms, from bacteria to ancient humans. Like, we have this huge library of genetic information now.
That's amazing. So we can read the DNA code super fast and accurately, but what if we want to look at a specific gene?
That's where DNA cloning comes in. It's like you're making photocopies of a specific page in that instruction manual so you can really study it.
Okay, so how do we do that? Do we need, like, tiny tweezers and microscopic scissors?
Not quite. We use bacteria and their plasmids, which are little pieces of DNA they carry.
Oh, right, plasmids. They're like these little bonus DNA circles.
Exactly. So scientists can put the gene they're interested in into a plasmid. Then they put that plasmid back into a bacterium. The bacteria replicates and makes copies of itself and the plasmid along the way.
So it's like we're hijacking the bacteria to make copies of the DNA we want. Pretty clever.
Yeah, it's a very efficient system, and this technique has changed everything from medical research to agriculture.
Okay, so that makes sense. But to put a gene into a plasmid, you'd have to cut and paste DNA, right?
Mm.
How do you even do that?
Ah, good question. That's where restriction enzymes come in. They're like molecular scissors that can cut DNA at very specific places.
So they're like tiny scalpels for DNA.
Exactly. And some of them leave these single-stranded overhangs when they cut, called sticky ends,
Sticky ends, why do they call that?
because they can bind with other DNA molecules that have matching overhangs. It's like puzzle pieces.
So the restriction enzyme cuts the DNA, and then these sticky ends help guide the pieces to reassemble.
Right. And then there's an enzyme called DNA ligase that acts like glue to seal the DNA backbone together.
Wow, it's amazing how everything fits together. What if you only have a tiny bit of DNA to start with, though? Can you still work with that?
Yeah, absolutely. That's where PCR comes in, which stands for polymerase chain reaction. With PCR, we can make billions of copies of a specific DNA sequence, even from a tiny sample.
Billions of copies from just a little sample. That sounds like magic.
It's pretty amazing. PCR uses this special enzyme called Taq polymerase, which can withstand high temperatures. The process involves these cycles of heating, cooling, and then extending the DNA strand. Each cycle doubles the amount of DNA you have.
Wow, that's incredible. So you're saying you could start with like a single drop of blood and get enough DNA to analyze?
Yep, that's right. PCR has revolutionized things like forensics, genetic testing, and even paleontology. Like, if researchers are working with old, degraded DNA samples, they can use PCR to make more copies.
So we can sequence DNA, clone genes, cut and paste them, and even amplify tiny amounts. What about figuring out what those genes actually do, though?
That's when things get really interesting. There are a bunch of different techniques for analyzing gene expression, like seeing which genes are turned on or off in different cells or situations.
So it's like figuring out which genes are active and which ones are quiet.
Exactly. One technique is called in situ hybridization. Basically, it uses these probes that bind to specific mRNA molecules, and then we can see where those genes are being expressed.
So it's like creating a map of gene activity.
You got it. We can see which genes are active in different tissues during development or even in response to environmental changes. It's really powerful for understanding how genes work.
This is all so fascinating, but what happens when there's something wrong with those instructions? Like, can we fix faulty genes?
That's a question that drives a lot of research right now, and we're finding that more and more we can. We're entering this era of gene editing with tools like CRISPR-Cas9 that let us modify genes very precisely.
CRISPR-Cas9. I think I've heard that name before. It's like a find and replace tool for DNA, right?
Yeah, that's a good way to put it. It's a system with the potential to cure genetic diseases, make crops better, and even control insects that carry diseases.
It's mind-blowing. But I imagine there are a lot of ethical considerations that come along with being able to manipulate DNA like that.
Oh, absolutely. This much power means we have to be really responsible. We need to have open discussions about the right way to use these technologies.
I agree completely. This deep dive is already blowing my mind. But we've just scratched the surface, haven't we? What else is in this chapter?
Well, now we can jump into the world of cloned organisms and stem cells. Remember how we talked about whether all the cells in an organism have the same genes?
Yeah, like, are the genes in my skin cells the same as the genes in my brain cells, even though they do completely different things?
Exactly. And scientists started exploring this idea through cloning organisms. Early experiments with plants showed some pretty amazing results. They took a differentiated cell from a carrot and were able to grow a whole new carrot plant from it.
Wait a minute, are you saying every cell in a carrot can become a whole carrot? That's crazy. What about animals, though? Can you clone animals from a single cell?
It's more complicated with animals, but yeah, it has been done. Dolly the sheep, for example, was the first mammal cloned from an adult cell. Scientists took the nucleus from a mammary gland cell and put it into an egg cell that had its nucleus removed.
So they basically swapped the DNA in the egg cell with DNA from an adult cell, and that made a whole new lamb.
Pretty much, yeah. That egg cell, with its new nucleus, developed into a lamb that was genetically identical to the sheep that donated the mammary gland cell.
That's wild.
Yeah.
But I've also heard that cloned animals aren't always exactly like the original. If they have the same DNA, how is that possible?
That's a great question, and it really shows how important epigenetics are. Epigenetics are the factors besides the DNA sequence itself that can affect how genes are expressed.
Epigenetics, I've heard of that. It's like adding these chemical tags to DNA that can turn genes on or off.
You got it. And it seems like the process of reprogramming a cell during cloning isn't always perfect. Sometimes those epigenetic tags can get messed up, and that can cause differences in cloned animals, and sometimes even health problems.
So it's not just the DNA sequence, but also these epigenetic factors that control how an organism develops. It's really interesting. So where do stem cells come into all of this?
Stem cells are like the ultimate blank slates in an organism. They have this incredible ability to both self-renew, meaning they can make more stem cells, and differentiate into all these specialized cell types.
Stem cells are definitely a hot topic in medicine these days. But honestly, I always get a bit lost with all the different types of stem cells.
It's understandable there are a few key types. First, you have embryonic stem cells. These are pluripotent, meaning they can become any cell type in the body.
Wow, so they can become anything. No wonder people are so excited about them.
Right. Then we have adult stem cells. They're a bit more limited in what they can become. They can only give rise to certain types of cells within a specific tissue.
Yeah.
For example, blood stem cells can become all the different types of blood cells, but not, say, skin cells.
Okay, so embryonic stem cells are like the all-purpose ones, and adult stem cells are more specialized.
That's a great way to put it. And then there's another type that's creating a lot of buzz. Induced pluripotent stem cells, or iPS cells.
Induced pluripotent, so they're made in a lab.
Yep, that's right. Scientists have figured out how to take fully differentiated adult cells, like skin cells, and turn them back into a pluripotent state, kind of like embryonic stem cells.
Wait, so you're telling me they can take an adult cell and basically rewind its developmental clock? How is that even possible?
It's pretty clever. By introducing a few key genes, scientists can basically reset the cell's epigenetic programming, making it pluripotent again.
That's amazing. What does that mean for the future, being able to create these iPS cells?
It has huge potential. Imagine being able to create stem cells that are a perfect match for a patient and then using them to repair damaged tissues or treat diseases like Parkinson's or diabetes.
It sounds like science fiction. I'm sure there's still challenges, though, both scientific and ethical ones.
Definitely. Working with stem cells, especially embryonic stem cells, brings up a lot of ethical questions. And even though iPS cells kind of avoid some of those ethical issues, there's still concerns about their safety and effectiveness that need to be addressed.
It's definitely a field that's moving fast with tons of potential and a lot of important questions to think about. So we've talked about manipulating cells and even cloning organisms. How is all this DNA technology being used in the real world outside of research labs? like in medicine, for example.
Get ready because DNA technology is changing medicine in ways we never could have imagined just a few decades ago.
Okay, I'm ready. Let's start with diagnostics. How is DNA technology changing the way we diagnose diseases?
It's having a huge impact. We can now identify hundreds of genetic disorders with PCR and DNA sequencing. We can even screen people for genetic predispositions to certain diseases before they show any symptoms.
So instead of waiting for a disease to develop, we can be proactive and maybe catch it early. That could really change things.
Exactly. It allows for early intervention and prevention, which can make a big difference for patients. DNA technology is also giving us a much deeper understanding of the genetic basis of diseases, even ones that aren't directly caused by a single gene, like cancer or Alzheimer's.
So even diseases that aren't considered strictly genetic can have a genetic component that we can target.
That's right. By looking at gene expression patterns in healthy and diseased tissues, we can identify potential targets for new treatments.
That's incredible. So DNA technology is revolutionizing both diagnostics and drug development. What about treating genetic diseases, though? Is it possible to actually fix faulty genes?
That's where gene therapy comes in. The basic idea is to introduce a healthy copy of a gene into cells that are missing it or have a defective copy. It's like replacing a broken part in a machine.
So it's like a genetic software update. How did that actually work?
One way is to use modified viruses as delivery vehicles. These viruses are engineered to carry the healthy gene into the target cells. It's already been really successful in treating some immune deficiencies, for example.
That's amazing. But I guess there are still some challenges with gene therapy, right?
Of course, getting the gene into the right cells and making sure it's expressed correctly can be tricky. And there are always concerns about potential side effects, like accidental mutations or immune reactions.
That makes sense. It seems like a field with a ton of potential, but it's still early days.
Yeah, but it's moving fast. And with CRISPR-Cas9 gene editing, things are getting even more exciting for gene therapy.
CRISPR again, it really is a game changer. How is it being used in gene therapy?
Well, unlike traditional gene therapy, which just adds a healthy copy of a gene, CRISPR lets us directly edit the faulty gene in a patient's cells. It's like having a molecular scalpel that can find and fix genetic errors with incredible precision.
So it's even more precise than traditional gene therapy. That's remarkable.
Exactly. And it opens up the possibility of actually curing genetic diseases instead of just managing the symptoms.
Wow. CRISPR really does seem revolutionary. But I imagine there are some really big ethical considerations when you start talking about manipulating the human genome like this.
You're absolutely right. We really need to have serious discussions about the potential consequences of changing the human genome, both for individuals and for future generations.
I completely agree. But before we dive too deep into those ethical questions, I want to hear about some of the other ways DNA technology is being used. We've talked about DNA technology in medicine, but I know it's being used in other areas too, like forensics, for example. I've heard that DNA evidence is a big deal for solving crimes these days.
Yeah, it's really changed things in forensics. Remember how we talked about PCR and its ability to amplify tiny amounts of DNA? That's super important for analyzing evidence from crime scenes.
So you're saying that even a little bit of blood or a hair can be enough to identify someone?
Oh, yeah, definitely. Forensic scientists look at specific genetic markers, these short sequences of DNA that are really different between people. They're called short tandem repeats, or STRs.
Okay, so by comparing these STR profiles, they can link suspects to crime scenes, or even clear someone who was wrongly convicted.
Exactly. It's incredibly powerful. DNA evidence is really important in our legal system now.
That's amazing how DNA technology is being used in all these different ways. I even read that they're using genetically engineered microorganisms to clean up the environment.
Yeah, that's right. It's called bioremediation. There are some bacteria that naturally break down pollutants, like heavy metals or toxic chemicals. We can use them to help clean up contaminated areas.
And we're basically using these tiny organisms to do the dirty work for us.
That's a good way to put it. And scientists are even making them better at it through genetic engineering. They can create bacteria that are super effective at getting rid of specific pollutants.
That's incredible. It seems like these microscopic creatures are doing all the heavy lifting. Speaking of using nature, we can't forget about agriculture. GMOs, or genetically modified organisms, are kind of a hot topic these days.
GMOs are definitely a big example of how DNA technology is changing the world. Scientists can add genes to crops that give them useful traits, like being resistant to pests or being able to tolerate herbicides.
So we're basically giving these crops a boost to help them survive in tough conditions.
Yeah, you could think of it that way. And it can have a really big impact on how much food we can produce, especially in areas that are dealing with climate change, water shortages, or other challenges.
GMOs seem like they could really help us feed a growing population, but I know there are concerns about potential risks too.
Yeah, definitely. Some people are worried that GMOs could harm the environment in ways we don't expect. It's really important to keep researching and monitoring these technologies carefully.
It sounds like a really complicated issue. We need to look at the potential benefits and the potential risks very carefully.
I agree. And it's important for scientists, policymakers, and the public to communicate openly so we can make smart decisions about these technologies.
Well, this deep dive has really opened my eyes to just how powerful DNA technology is. And it's not always simple, but it's definitely fascinating. It's incredible to think that we've gone from figuring out the structure of DNA to actually being able to manipulate it in all these different ways.
It really shows what we can do when we put our minds to it. And who knows what amazing things we'll discover as we learn even more about DNA.
That's what's so great about science, isn't it? It's this never-ending journey of discovery. And the more we learn, the more questions we have.
I couldn't have said it better myself. That's what makes these deep dives so interesting.
I completely agree. Thanks for joining me on this exploration of DNA technology. And to everyone listening, thanks for diving in with us. Until next time, keep those minds curious.